Okay, so we're talking about the central dogma of biology. And we had said, what did we say was the function of the nucleus? Control center of the cell. And yet, when we talk about the what do genes do, nothing but proteins. But we know that there's lipids, there's nucleic acids, carbohydrates. So what about those? We'd said the nucleus is controlling everything, and yet when we look at what does a gene do, it makes a protein, or it codes for the information that is used to build a protein. So what about those lipids? Well, it involves proteins. One of the major types of proteins was what? We're on slide 15. Enzymes. Proteins as enzymes control chemical reactions. So the slide that we're looking at here has to do with making testosterone. It's a steroid hormone, right? And it's controlled by a protein. So proteins as enzymes control all that other stuff. So the nucleus really and truly does control all of the activities of the cell. Okay, so different cells express different genes, and so they do different stuff. And so the different enzymes that they have coded for then determine what that cell is going to do. And it's why a bone cell makes bone, and a muscle cell produces movement. And they all have the exact same genes, but they turn on and they express different genes. All right, so we know a little bit about the human genome because in 2003, they finished a study where they sequenced the entire human genome. And what did we say we found? They found in those 23 pair of chromosomes, 35,000 different genes. But then we now know that those 35,000 different genes can code for multiple polypeptides, multiple proteins each. How? Alternative splicing. Very good. You guys are great. I'm assuming some of you got it. I hope so. I'm really sad if not. But anyway, alternative splicing of that messenger RNA to produce those different mature messenger RNAs. And so then, as a result of that, we can explain the 90 some odd thousand different proteins that are expressed in humans. Now, most of those chromosomes, we said we got those 23 pair of chromosomes, and they're gigantic molecules. We're almost all absolutely identical. We're 99.99% identical, but then there are those little differences that make you look like me instead of like you. And so as a result of that, even if we don't know about your genes or my genes, we know about the genes that are controlling stuff. So they vary between you and me, but there's a lot of similarity. And so as a result of that, this Human Genome Project allows scientists to move into this new field of sort of gene therapy type treatments. And one of my wife's biggest criticisms of the cancer research is that drug companies, they love, well, that's really not the right term, but they make a lot of money off of the fact that cancers will mutate very readily. So you have different, what they call phenotypes of a cancer, and you can make a drug. They say, oh, this is such and such phenotype, and so you say, we got a drug to take care of that. And then so they sell the drug and they treat the person, and the person is getting better, and then they have a relapse. And what's happened is the cancer has evolved, has mutated into a different form, and the drug companies say, we've got a treatment for that, and so they can sell lots and lots of different drugs. Eventually, unfortunately, with cancers, typically you run out of, they say, that one, we don't have any treatment for it. So that was why with her research, she was so excited about it. It didn't have to do with the different phenotypes of the cancer, which is an outgrowth of this Human Genome Project, but it was because of that heat shock 90. It was like this thing that would treat many different phenotypes. But anyway, medicine moves along, and right now there isn't a whole lot of money in medical research, but hopefully the pendulum will swing back to where more money goes into it. But anyway, so hopefully you understand how genes are expressed, and we'll talk about it on Monday. All right, you guys have a good day.